Thank you very much for the introduction. Good afternoon, everyone. Apparently, my job is to wake up everybody. So I have a presentation very different from what you heard since like morning, amazing talks. And to be honest, I'm a little bit shaky because I'm an engineer. I do like engineering stuff. I work on like making tools. And again, my passion is in diagnosis. So definitely, I have very good friends and collaborators at hospital, and I tried to learn the language of how to communicate because engineering, or let's say science, is very much, again, in another word. You talk about some definitely different type of technologies rather than, let's say, clinicals. So, but what we do in terms of diagnostics and what we mean about liquid biopsy in what we're doing in liquid biopsy. Again, I cannot really talk about, let's say, the core medicine or biology, but at least what we learned in liquid biopsy is try to understand the state of the disease by looking at biomarkers that we engineers always appreciate simplicity. All these biomarkers are freely available to us through body fluids, through blood, urine, any kind of body fluids that you can imagine. So this biomarker could be varied from from vesicles, from nucleic-based biomarkers. We have great talk on ctDNAs. They can be RNA-based, protein-based. But in particular, what I'm going to talk in, today is one of very, let's say, particular technologies that we developed to tackle extracellular vesicles at the single level. But why we are doing this diagnosis, what at the point of care or at the point of need. So definitely in terms of liquid biopsy, there are many techniques, standard techniques that are there available in centralized facility that they mostly based on lengthy protocols. They are mostly basically dependent on the basically highly qualified personnel, and definitely not very much user-friendly in terms of operation. We cannot easily bring it to home. We cannot bring it necessary to remote clinics. So that's why we define devices that we say they are usable at the point of care or at the point of need. And these devices potentially can achieve the same accuracy and basically sensitivity, whatever keyword that you want to basically bring here, the accuracy of the basically a standard test, and potentially can be also automated. Let's say we don't necessarily need to go to facilities. We don't need to go to hospitals. So we can really use it any place. Sorry, I just hear that I probably have to keep this. And potentially we can also reduce the price of these devices, again, by bringing engineering approaches, automation approaches. So these kind of devices are basically what we call microfluidic or miniaturized based devices that can combine all these basically keywords and elements. And of course, the race in engineering side, in science side, is how to make these devices in a way that you like it. Yeah, it can basically bring to the hand of clinician or people in need to efficiently run and really like try to reduce the time of action on diagnosis, prognosis, and even monitoring therapeutic. So in my lab, we basically combine a couple of approaches. Then one of them, as was introduced in terms of nano bio devices, is bringing the enhanced properties of what we call nanomaterial. Probably you have heard about nano. It's not new. Anything between 1 nanometer, 100 nanometer is called nano. But what is important here for us, it's that exciting properties of these kind of materials. For example, if you consider gold, gold is shiny, is beautiful. When it comes to nanoscale, it has some interesting phenomena or properties that can be brought into sensing application. So there are different type of material that what we call bring enhanced properties from electrical, optical. So if you assume a device for, let's say, sensing, optical, colorimetric device, for example, dipstick device, a glucometer-based device, all these material can play an important role in enhancing the sensitivity. But definitely you cannot just hold this material in hand and let's say, run any assay unless they are combined with a sample handling system. That we call microfluidic. So these microfluidic are great in terms of bringing those lengthy protocols, in terms of bringing, let's say, the a large sample volume in a small scale. And at the end of the day, our job is not complete until we can automate the whole package. So basically these three are, I should basically also add another circle here about using AI that help us to do data analysis much more rapidly. So basically these three items are the core of research what we do in the lab. And these are example of devices that we develop that bring all these key elements of science and engineering into a benchtop tool. Of course, we publish, we have patents, more than 7, 8. We have started commercializing a couple of these technology, handheld or even benchtop tools in the lab. But in particular, one of a very new, let's say, technologies that we are looking at, and I hope with that I can attract your attention today, is how we can bring these devices into application for extracellular vesicles. So extracellular vesicles is a kind of, I even believe in your word, is a new biomarker for even cancer or any other disease. But why we are interested, again, from engineering side point of view to these biomarkers, because there are single vesicle lipid particles that carrying the biocargo. Assume that they carrying the biocargo, their surface protein. So definitely it's a valuable biomarker shed from cells, again, freely available to us in the body fluid. So we can, if we access this basically information that buried into these biomarkers, then probably can achieve many valuable, let's say, information about the let's say diseases, again, like cancer. Even there are sign of there are research now on EVs for infectious disease, but in particular here our attention and our interest was trying to see how we can access EVs for basically researching cancer. But one of the problem of EVs when it comes to centralized facility and tools available in central facilities is heterogeneous. So they are basically they are usually assessed in a pool of population of EVs. So the access to information at the level of single EVs, which are very usually in a low concentration, it's very much valuable in terms of let's say, clinical studies. So it's basically a very non-traditional, what I'm going to talk about right now, very non-traditional way of looking at EVs. So what we develop in terms of tackling EVs is basically a very a nano array of a platform that still use these smart or what I say, the magic of nanomaterial, smart nanomaterial. So by using this platform and capturing EVs at the level of the cavities, we can fingerprint them by using what we call Raman scattering. So Raman scattering has been there for so long. Basically, it's a method that can transfer the radiation, basically the vibrational and structural mode of molecules and chemicals into a spectral peak. But what happens when we use nano material? We can amplify, again, because of the magic of the material and enhanced properties, we can magnify these scatters. So if you look at basically the difference of these two, let's say, peaks, that one is based off advancement of surface enhanced Raman spectroscopy, and a regular one, you will see that in this spectral coming from this surface enhanced platform, which we call Mosers, is much more amplified. It can gives us more information. Now, how we can use it for cancer? If you look at these different basically plots that or spectral peaks in this plot, you will see that if you look at, let's say, just a pure lipid bilayer, or if you Raman, let's say, a non-cancerous, let's say, healthy cell line versus a cancerous cell line here, definitely their fingerprint is different with Mosers. Even with the PCA analysis, we can exactly show that these fingerprint from cancer cell line to basically non-cancer cell line and pure liposome is absolutely different. Even one step further, we could basically identify in the level of cell lines that we can differentiate between a parental, for example, U373 and U73 carrying, let's say, EGFR variant 3. Again, even in terms of, let's say, an amplified gene such as EGFR variant 3, we can still identify this structural peak. But how we can use this? I mean, it doesn't mean anything until we really test with human sample. It was then when I basically met my very good now friend, collaborator, Dr. Ganesh Rak, and later on Livia Garcia, that why not we can bring this, let's try if we can bring it to any, let's say, clinical validation. Again, I don't call it clinical study, I don't call it clinical trial, just validation and understanding whether we can do anything with this platform. So again, we work with a library of GBM cell lines, characterized cell lines. We collected all these spectral peaks that represent, let's say, single EV average. We did all these, let's say, PCA analysis that now I laugh at because it took us, let's say, couple of just weeks to analyze. It was then that we thought, okay, let's bring machine learning in. So machine learning help us to just do this data analysis in a more harmonized and, of course, accelerated way. So after basically we spend like we try to develop a machine learning algorithm that can help us to extract information from this spectral peak to do the binary classification between healthy patient and also understanding the molecular alteration. So again, we continue to train our machine learning algorithm by looking at a variety of cell line. We use GBM as our model, a variety of cell line from GBM, and understanding how we can do the accuracy measurement along this cell lines. This we use as a library, as a trained algorithm library to basically look at the patient data. We had access through MNI to 12 GBM patients and trying to understand whether we can classify within the basically cancerous and patients, and more advanced, whether we can classify between the different, let's say, molecular alterations, if we're here, EGFR, variant 3, and MGMT. Again, we used MB as our model. We used the same training of cell lines on MB cell lines and two different basically classification between G3 and G4, and moreover patient studies, like we did this validation also on 23 MB patient sample, binary classification and classification between G3, G4. But at this point, you probably see what does all mean here. These spectral, of course, we can classify, but can we also correlate with any of these genomics or proteomics standard technique? This is exactly what we are doing right now. We are trying to correlate our spectral peak with and understand how this classification we are doing can basically achieve the same accuracy of sequencing, the same accuracy of proteomics studies. Basically, the last slide of my talk, definitely I don't want to give any perspective of where we are going. It is still in the discovery, no perspective of what will be the future. But this is just a very small, a quick overview of a very completely non-traditional technology that can advance up relatively good sensitivity in terms of, let's say, accurate diagnosis, and bring the time of diagnosis relatively low. Like scanning this platform is basically taking about maximum 1 hour. And again, because of the simplicity of designing, we can easily, this is what we are now going through, to combine this with a microfluidic and a benchtop tool, so we don't need to use a, let's say, Raman spectroscopy sitting in a research lab anymore. Of course, we also have challenges in terms of manufacturing, scaling, and throughput that we are trying to achieve. At the end, I would very much like to acknowledge the funding agencies and also all my collaborators at hospital and also science to achieve, and all my lab members, students, and definitely these three little, for my student and myself, lab members. Thank you.